Bom dia a todos. Vamos começar mais um seminário aqui do ciclo de seminários da pós-graduação do Instituto de Biologia da Unicamp. Hoje a gente vai ter uma palestra muito interessante da uma pesquisadora, professora da Universidade da República em Montevideo, no Uruguai. Lembrando que estes seminários são uma combinação entre 3 disciplinas da pós-graduação do Instituto de Biologia da Unicamp. E ao final da aula, todas as perguntas devem ser feitas aqui no chat, no YouTube. Tá? Então, lembrando que para vocês se inscreverem, para vocês fazerem perguntas aqui no nosso chat, você deve estar inscrito no nosso canal. Tá? Então, eu convido agora a apresentação, a doutora Mrna do Agon. Bom dia.
Bom dia.
Doutora Mrna, eu agradeço publicamente a sua presença, a sua disponibilidade de estar aqui conversando com a gente hoje. A doutora Mrna é bióloga, fez graduação no Uruguai, né, na Universidade da República, onde fez o mestrado também, né, doutorado também, e depois, você quer contar um pouquinho dessa história, Mrna, por favor?
Eu vou começar, bom dia, agradecendo também sua invitação a participar aqui. Eu nunca aprendi português, somente estou falando e criando uma linguagem que conosco porque adoro, me gusta muito do Brasil, mas desculpem porque é muito ruim. Então,
tá ótimo.
Vou
Tá
trocar para inglês depois, porque acho que você me vai compreender melhor. Então, eu estudei na Universidade da República, em Uruguai, eu sempre trabalhei lá. Eu fiz um doutorado sanduíche com a Universidade Estatal de New York, a Buffalo, estudando a regulação da expressão gênica no parasito Trypanosoma cruzi. Eu sempre trabalhei também, paralelamente, em genética humana, estudando enfermedades congénitas. ¿sí? Sempre buscando mutações e cambios em proteínas particularmente reguladoras da expressão gênica. Sempre meu interesse se concentrou na regulação da expressão gênica. Trabalhei também na Cleveland Clinic Foundation, em Cleveland, estudando também fatores de transcrição que regulam a expressão gênica em cardiopatias congênitas ou em famílias uruguaias. E agora tenho um grupo de trabalho próprio. Quando retornei do pós-doutorado no Instituto Nacional de Câncer, eu trabalhei em cancer stem cells, ou células progenitoras, células tronco de câncer, e retornei ao Uruguai e comecei uma linha própria de investigação em ARNs não codificantes em câncer. E vou falar a vocês de parte desta investigação.
Perfeito, ótimo. Muito obrigado. Então, agradeço de novo a sua disponibilidade, e eu sou suspeito para falar, mas o tema dessa palestra hoje realmente muito instigante, né, e acho que a doutora Mrna vai mostrar um pouquinho do que é o trabalho justamente investigativo, né, de tentar descobrir a natureza, o papel biológico de uma molécula de RNA não codificador. Tá bom? Então, eu vou compartilhar a sua tela aqui.
Ok.
Então, doutora, boa aula. Obrigado de novo.
Murilo, tenho uma hora máximo, né?
É isso.
Ok.
Perfeito.
Muito bem.
Ok.
Obrigado.
Boa aula. Tchau, tchau.
Ciao, ciao. Then now I'm going to switch to English. I hope you understand me more than before. So the title of the talk, it's To Be or Not to Be, the non-coding RNA 886 Intriguing Nature. So what I want to talk about is in a real story about how we have been switching our research goal because of the revolution that has been happening in the non-coding RNA field. Sorry, some of the slides are in English, some others are still with parts in Spanish, but Spanish is similar to Portuguese in many ways, so I hope you will understand the Spanish also. Sorry. So my work is in prostate cancer mainly. Just a little bit of introduction. For an introduction, is the most frequently diagnosed cancer in 105 countries, the second in other countries, and is the most common malignant tumor in men. Actually, 1 in 6 men will be diagnosed with prostate cancer in their lifetime. So this is statistics from GloboCan 2018, and you can see prostate cancer here. Fortunately, the mortality of prostate cancer is low compared to, for instance, lung and other cancers. So this is a distribution of the incidence of prostate cancer worldwide. And here is Brazil. As you can see, there is an even more incidence than in Uruguay, and similar to most of the first world countries. Another thing that I would like to tell you, thinking that maybe some of you or most of you have or do not belong to the cancer field, is some general concepts of cancer. So here is a typical presentation of the cancer hallmarks. There's a gene. This is the androgen receptor, a very important oncogene in prostate cancer. But whatever the gene is here, cancer affects many cellular attributes. Those can be cell death, can be cell proliferation, can be cell interaction with the immune system, DNA repair, metabolism, and angiogenesis or neovascularization. So cancer revolutionized the cell in many different ways, affecting several cell phenotypes. One gene can also affect more than one of the cancer phenotypes. So mutated genes in cancer usually contribute not only to one, but to many aspects of the cancer biology. Some of them the gene can promote, while others the gene can suppress. And this is a particular representation of the effects of the androgen receptors, promoting proliferation and inhibiting apoptosis. So these are the hallmarks of cancer. Cancer is a multiple gene mutation disease. It requires usually several mutations between 1 and 10, and usually 5 in solid tumors. Those mutations are the drivers of carcinogenesis. The effect of a mutation in a cancer gene could be the up and down regulation of the gene, meaning that the gene remains perfectly in nucleotide sequence, but the number of transcripts for the gene are higher or lower. Or what we see in other genes is a gain or loss of function. And that happens usually affecting the protein activity, and that usually depends on mutations in the nucleotide sequence of the gene. So within both mutational mechanisms produces changes in the gene expression of the cell that will end up in the promotion of the cell transformation. Cancer genes are classified in 3 main groups. One is tumor suppressor genes, the other is oncogenes, and the third is caretaker genes. So tumor suppressor genes are genes that normally in the cells promote cell survival, cell proliferation, cell movement. Whereas that's for oncogenes. Tumor suppression genes are actually the opposite. Those in the normal cell physiology, those genes break or stop or reduce cell proliferation, and are those genes that the cancer usually would like to kill or block or mutate to disrupt the functions. Whereas for the oncogenes, cancer cells, what usually acquires it's an overexpression or an overactivation of those. So caretaker genes are genes that are mostly involved in DNA repair and cell cycle control. So this story begins in 2010 when I was at the National Cancer Institute investigating cancer stem cells. So look here, sorry, this in this part of the slide, I'm showing you that a model of cancer stem cells that we developed at the National Cancer Institute. So this is a way to isolate stem cells in a cancer tissue or in a tissue culture of a cell line, in which you can the cancer stem cells grow as spheres in a particular media. So we did a characterization of these spheres. I'm not going to go in detail on that, but I want to tell you that we were studying the differentiation from these cancer stem cells to more differentiated cells that are less aggressive cells. And you see that the aggressiveness of the spheres versus the differentiated cells is much higher. Also, the expression of stem cell markers, characteristics of the stem cells, embryonic stem cells and adult stem cells is increased in the spheres. So what I did was a screening of microRNAs that were expressed in the spheres and then expressed after the spheres were induced to differentiate into adherent cells. So I isolated small RNAs and I analyzed the expression using an microarrays from Exequon. This was this company specialized in microRNA products. Among the differentially expressed microRNAs, I found this one called HSA Mir 8863P. And this is the subject of the study. I would like also to give you this message. Most of our genome is composed of non-coding RNA. Take a look at the small fraction of protein coding genes we have, only 1.5%. The rest of the genome is non-coding genome, mostly actually composed by repetitive sequence, but there's a huge proportion composed of introns. So our last question in the genome study field is, what is the 98% of the genome doing? The one that 98.5% that is not coding for proteins. And this has been also called the dark matter of the genome. And most of the efforts in the genomic field are currently focused on this dark matter of the genome. The other interesting thing that we recently discovered is almost the entire human genome can be transcribed. And that was a very big surprise for people. So why is everything transcribed? What's on the dark side of the genome that needs to be transcribed? Does it play any role? So in the field of non-coding RNAs, many hallmarks had been happening. This is a timeline with showing where the most important discoveries in the RNA field. So I'm not going to go into detail, but I want to tell you that in 1993, the microRNAs were firstly discovered. And then the field has been growing very rapidly. More recently, the other field that exploded is the field of the long non-coding RNAs or links or LNC RNAs, and other type of RNAs are currently flourishing. This has been also motivated or stimulated by the enormous technological development in the sequencing field. And just to make you see the rapid evolution of the field, see that in microarray field is born between around the 2000s, and the transcriptome, the transcription, the RNA-seq field, 10 years later. So my project started in between with the first microRNAs arrays that were developed by Exequon. So that was about here. During that time, the transcriptome sequencing of small RNAs was started to develop. So new data came out after I started this project, and this new data was transcriptomic data, but it took a longer time to be published. So this is like in my lifetime, the many changes that I've seen. RNAs, non-coding RNAs, are classified in 3 main classes depending on the size of the RNA. Small RNAs, mid-size RNAs, and long RNAs. 2 to 35 nucleotides are considered small, 30 to 200 nucleotides, middle-size RNAs, and 200 and over long RNAs. In the group of small RNAs, I think you know many of them, like small interfering RNAs, PIWI RNAs, PIWI-associated RNAs, RASI, repetitive element-derived RNA, microRNA, the subset of this, and whatever small RNA derived elsewhere is going to have this acronym small derived something, deriving from somewhere. In the middle size group of RNAs, you have small nucleolar, small nuclear, transfer RNAs, the bulk RNAs, Y, prompt, TSAs, and TSL, and many, many others. Then for long RNA, there's also a lot of different types, and again, I don't have time to go in detail. So our talk involves microRNA and bulk RNA, the research that I'm going to tell you soon. For those who haven't seen what microRNA are, microRNAs are small RNAs of 20-22 nucleotides long. They are coded in our genome as single genes or inside the introns of protein coding genes. They are processed by DROSA or by the splicing machinery to form short hairpins that needs to be exported to the cytosol. And in the cytosol, they are processed by the RNA-induced silencing complex, the same that works for small interfering RNA. And then they are processed to small double-stranded RNAs of the 122 nucleotide length. One of them is going to work as a silencer of gene expression. The way they work is by pairing in a complementary way with the mRNAs of protein coding genes, leading to either the cleavage and degradation of the mRNA or the repression of its translation. So in both ways, what a microRNA is going to do is to reduce the expression of the protein, the production of the protein. All of these is mediated by the RISC complex, in which the argonaute proteins are essential components. Other small RNAs are coded and processed in completely different ways, and they are involved in totally different mechanisms of action. So if you see the structure of a precursor of microRNA, I just wanted to show you that they are usually hairpins with a double-stranded regions, the RNA pairs following Watson and Crick rules, and then there is a regions of bulges or a symmetrical or asymmetrical loops. MicroRNAs then interact with this is a microRNA with the messenger RNAs through a small region of 7 nucleotides, and the second to the 7th position, 6 nucleotide canonically, that has perfect complementarity to the mRNA, usually the 3' UTR. But the rest of the microRNA does not need to complement to the target sequence. MicroRNAs also regulate more than one gene, more than one messenger RNA at the same time, and they usually are fine modulators of gene expression. They produce small changes in the expression of the proteins. In cancer, microRNAs can be tumor suppressors, can be oncogenes, with the same rationale than the one I mentioned before. Tumor suppressor RNAs will actually, in the normal cell, contribute to the restraint of the cell cycle and the invasiveness of the cells, whereas the oncogenic microRNAs in the cell usually are promoters of proliferation and cancer hallmarks. So what we will see in cancer mostly is that oncogenic microRNAs are upregulated. There's more expression of the oncogenic microRNAs in cancer, and that will produce the repression of tumor suppressor genes, tumor suppression proteins. In cancer, also, what we see is the downregulation of tumor suppressor microRNAs that will lead to the overexpression because of a release in the repression of oncogenic proteins. In that way, microRNAs contribute to the development, maintenance, and evolution of the neoplasia cancer-wide. Okay, this is the slide that I showed you before with the proper animation. So here is the same thing. And these are the articles you can read that we published on the cancer stem cell model. I thought that the previous one was the wrong one. I'm not sure why it remained on the presentation. So I will go faster. I already showed you that. Why this RNA is called HSA microRNA Mir886, the one we are studying, the focus of this investigation. That means Homo sapiens microRNA precursor 886. So this is the nucleotide sequence and the secondary structure of the microRNA, this particular one. And you see the hairpin and the bugs here. This is just box A and B are a part of the promoter of the RNA polymerase 3, because this gene is transcribed by RNA polymerase 3. And if you see, this precursor was predicted to generate 2 microRNAs, one from the 3' end, and this is called microRNA 886-3P in purple, and from the other complementary strand, the complementary microRNA called Mir886-5P here, okay? So that was our idea when we started working with this microRNA. We isolated as a differentially expressed microRNA on microarrays. We did also qPCR to confirm the downregulation of this putative tumor suppressor microRNA because it was upregulated in the differentiated cells. And then we started to do some preliminary functional experiment, transfecting, overexpressing, blocking this nice, new, and unstudied microRNA at that time. However, then there was one student starting this project. It's called Rafael Ford. I will show you his picture. It's connected now. He began his graduate thesis in this project. So actually the postgraduate thesis was in this project. So one day, starting with this, we wrote this project, microRNAs, and 886, 3P was the one we identified. And we've designed a strategy to study this microRNA in all the possible ways that were used at that time. And we got highly surprised by this finding. Mrna is the repository of microRNAs, where all the microRNAs are listed, cataloged, and annotated and curated. So in 2010, HSA Mir886 was eliminated from Mirbase. And this sign was in Mirbase said death microRNA entry. With this comment, the annotated mature sequence of this fragment of this microRNA is actually a fragment of the volt RNA. And read the reference if you don't understand what that means. When we read that, we said, oh, come on, volt RNA? What's that? I don't know if you have ever heard about the volt RNAs, but we didn't at that time. So we needed to read the paper by Stallard et al. What is a volt RNA? I'm sure you know what a ribosomal RNA, tRNA, ciRNA, microRNA, but surely not volt RNA. So this is the article when this is the evolutionary biologist looking at the conservation of sequences along the evolution of species. And this group found that actually this microRNA precursor that was initially annotated as microRNA precursor had a very strong homology with the volt RNAs. So here are the 4 volt RNAs. In 2010, there were only 3 volt RNAs annotated in the human genome, 1.1, 1.2, and 1.3, and this is an alignment of them. And this study, for the first time, proposed that this precursor of microRNA was indeed a volt RNA. And the evidence was absolutely compelling, strong, and robust. So no doubt that the origin of this molecule was the volt RNA. Well, let's see what's a volt RNA, what a volt RNA is. So volt RNAs are RNAs associated to the volt particle. Sorry, this is in Spanish. Volt means, in English, if you translate this structure, like where would you put wine or water, and the volt RNAs are actually composed by, first of all, they are the biggest ribonucleotide particle in the cell. They are bigger than the ribosome. It's a very nice particle. Look at the electron microscopy sections and also the draws of the particle. They are cytoplasmic particles composed by the measurable protein mostly, TEP1, PARP1, and volt RNAs at the top of the structure. I'm not going to go into in detail, but here is the volt RNAs location in the particle, and this is the structure of it. This is volt RNA 1.1, 1.2, and 1.3. As you see that they share A, B box and structure and sequence. And then they are actually very, very old in the evolution time. They are present from early deuterostomates, but we don't know much about the function. And that's something that is why nobody knows much about volt RNAs, mystery. So this is volt RNA 2, the one we were studying is not 1, 2, 3, but it's a more recent emerged copy of the volt RNAs. It's similar, but it's different at the same time. It's longer. It has it's codon in Spanish, hinge or something. It's similar, but it's not exactly the same, and it's actually located in farther away than the volt RNA 1 cluster. So we thought at that time, well, maybe this is not a microRNA precursor per se, but maybe it is also capable to produce RNAs, small RNAs, by who knows what. But those small RNAs may be able to repress gene expression in a similar way than the canonical microRNA. And that is part of our story. So the two goals that I'm going to show you quickly now was that we wanted to pursue were for the first time to understand if this molecule in prostate cancer act as a big volt-RNA precursor, medium-sized volt-RNA precursor, or as a small RNA, or if it acts as both, and if it has any implications in the cancer biology. So the general aim was to discover the implication of the deregulation of volt-RNA 2.1. I forgot to tell you that not only it was initially annotated as a microRNA precursor and later as a volt-RNA, but more recently, since there's a lot of debate on its function, some people decided to name it as a non-coding RNA 886. So in the literature, you will see 3 different ways to call the nomination of this volt-RNA as microRNA precursor, canonical microRNA precursor, volt-RNA 2.1, and non-coding RNA 886. So we wanted to see confirm if it's deregulated in cancer, in prostate cancer in particular, if it's capable to produce a small RNA, and if either the whole volt-RNA or the small RNA derived from it potentially were affecting the phenotype of the cancer cell. So for the first goal, we wanted to determine the implication of the deregulation of the precursor in the origin and maintenance of the tumor phenotype of prostate adenocarcinoma. Yes, as a second goal, we wanted to determine whether volt-RNA 2.1 non-coding 886 serves a precursor for the formation of small non-coding RNAs and assess the implications of their regulation in the origin and maintenance of the tumor phenotype of prostate adenocarcinoma. I will also show you just the link to go to a more recent article we have as a preprint that is about the global expression of volt-RNAs in cancer, but that's a third thing. So these are the 2 main goals. The first one is this one. We wanted to study the volt-RNA or non-coding RNA as a whole molecule. For that, what we did was we used lentiviral vectors with the full sequence of volt-RNA 2.1 non-coding 886 and a control RNA with a similar structure but a completely different sequence not present in the human genome. So with these lentiviral constructs, we transduced cell lines, prostate cancer cell lines cultured in vitro in the laboratory. These 2 are representative of the androgen-insensitive prostate cancer and the androgen-sensitive prostate cancer. So with these lentiviral vectors, we were able to overexpress this precursor. So you can see that this construct has also a M-cherry reporter. So you can see the expression of the reporter, meaning that the vector was inserted in the cell. It was selected by, I think, puromycin. We also did the transfection of the small RNAs for the second goal, but that was a transient transfection. This was a stable transduction, and this is a transient transfection. With transient transfection of the small RNAs, here we call them small non-coding 886 3P RNA, small non-coding RNA, instead of microRNA, because since it derives from a volt-RNA, we decided to be more neutral and don't call it a microRNA because it derives from a different, not a microRNA canonical precursor. So with this technique, we overexpressed the microRNA in the same cell lines and others. We used QIAGEN technology for microRNAs. So with these two different gain of functions, we actually looked at different cell phenotypes, like cell viability by MTT, cell psychoanalysis by propylium iodide, annexin 5 to look at apoptosis, matrigel invasion chambers, Boyden chambers with for assessing invasion capacity. Also, to look at the changes in gene expression induced by these molecules, we used a microarrays, gene expression microarrays from Affymetrix. And then we did in vivo experiments for this Volt RNA study in nude mice. Then there's a part of the study that is a bioinformatics study, that is the analysis of the expression of this microRNA in transcriptomic studies. Remember that this microRNA was removed from Mrna. So if you want to look at the different transcriptomic studies for small RNAs, if you want to study the expression of this particular small RNA, that is not anymore a microRNA, is not considered anymore a microRNA, you don't see any annotated reads for this microRNA. Usually the microRNA files, annotated files, include all the annotated microRNAs in Mrna, and this was removed, so it's not there anymore. So we needed to start with the raw data of reads to find the reads belonging to mapping to the microRNA precursor and to this region, the 3P region. So here is a different software and algorithms that we used, and we look at a lot of data that is published on cancer, mostly from the Cancer Genome Atlas, and we reanalyze the small transcriptomic data to find reads for the small RNAs derived from the Volt RNA 2.1. The most important data set that we used was the Cancer Genome Atlas. The Cancer Genome Atlas has the richest amount of information, genomic information in every pathology, and it's the omics information is at several levels: mutation, copy number, gene expression, DNA methylation, microarray, clinical data. And for prostate cancer, we have available for around 550 samples of different patients, including normal and tumor tissue. So it's an amazing repository, and it has also developed a lot of tools to query, to investigate the different levels of data in simple ways. Then we also look at all the expression in cancer, but that's not the subject of this. So in the first goal, I think to go quicker because this is published data, and I think I took a little bit of time introducing many concepts that are actually needed to understand this. So since the papers are published, I'm going to be quick, and I will give you the slides of the presentation so you can take your time to look a bit more slowly on the details if you want. So I'm going to show you just the big messages. So here what we found, firstly, analyzing the Cancer Genome Atlas data, DNA methylation data in all these cohort, the big cohort of around 500 patients and normal tissues, what we saw is that the Volt RNA 2.1 or non-codinated SIG gene is strongly regulated by methylation. And what happens is that a DNA methylation at the promoter region inhibits translation. So what we saw was in that if we look at the DNA methylation in normal versus tumor tissue, in the TCGA cohort and Stanford cohort, you see an increase in the methylation in the tumor tissue. in both, a significant increase. And that is also seen when you compare normal to metastasis. So that means that the gene is silenced probably by methylation after the malignant transformation. We also see that the methylation of the promoter of the gene is associated to several clinical values of aggressiveness, including the Gleason score, that is a histopathological classification system, graduation system for prostate cancer, the clinical t-value, that is a general scoring system in cancer, and the biochemical recurrence, meaning that the prostate-specific antigen increase. So these three clinical variables are associated to the levels of methylation, therefore the levels of expression of the RNA 2.1. We also look at the gene expression, the methylation of the promoter region. These are CpG island, the ones that we studied at the promoter, in normal and tumor tissue, in both cohorts in more detail. And what we saw was there's a a very important variation in the methylation of these promoters heterogeneity in the patient. As you see, there are normal people with low methylation and normal people with high methylation initially of the promoter, but no matter what is the initial state, even though it's polymorphic, what we see is always an increase in the methylation in the matched tumor tissue. And that has been a very, very important challenge in this project, because RNA 2.1 turned out to be one of the most polymorphically methylated locus in the human genome. And that was a puzzle for us, because we saw that at the very beginning of the project, and we didn't understand why there was so much variation in the normal DNA methylation in the human population. And then many papers have been published recognizing this surprising feature of this loci, which means that probably it is very important. So meaning that many individuals have low levels of methylation, others have high level of methylation, but no matter what, in the tumoral cells, there's going to be this is the methylation here, an increase in the methylation of the gene. If this is one individual, this is a second one, normal and tumor, normal and tumor. And if you look at the expression of the RNA, what is expected is that this increase in methylation will lead to a decrease in the expression of the gene. Again, even though the patients have different levels of methylation and expression, there's usually a decrease in the expression in comparison to the initial level in the normal tissue. That's very, very unusual. But we saw that, and other people have later on seen that in other tissues too. Same situation happens on the cell lines. So these are different prostate cancer cell lines, normal and neoplastic cell lines, these 2 and 3. And we see there are changes in the methylation that are correlated with the changes, inverted changes in the expression of the RNA 2.1. So in the cell lines, the same phenomenon that we have seen in patients is happening. So cell lines are good models to study this gene. So we demethylated by 5-ACE-cytidine the cell lines, and we saw an increase in the expression of the precursor, meaning that the methylation is regulating the expression of the transcript. We also have a small Uruguayan cohort of patient tumors, prostate tumors, and what we saw is that in our tumors, there's also a decrease in the expression of this gene in malignant tissue. And then we actually found out that probably DNA methyltransferase 3B is a candidate regulator of this methylation, this change, this increase in the methylation in the tumor tissue. But I'm not going to talk much about it. It's in the article if you are interested in. And when we looked at the phenotype produced by the RNA in the cell lines, we saw that the overexpression of this non-coding RNA, Volt RNA 2.1 or non-coding 886, from there on, there's a decrease in cell viability. Also, if there's a change in the distribution of cell cycle in vitro in the cell population, that if you can see there's an increase in G2 M phase, and also the cells lose invasive capability in the two cell lines. We also did some experiments in vivo by subcutaneous xenotransplant of the VU145 cell line transduced with the lentiviral constructs, and we found the same effect in a reduction in proliferation in those cells that are overexpressing non-coding 886. And when we did the pathology of the little mice tumors, we found also a decrease in average tumor weight, the number of mitosis, and the percentage of necrosis. So these assays also confirm the proliferative effect of non-coding 886 seen in vitro in the cell lines. We finally wanted to see if this proliferative effect that we have seen in the tissues, in the cell lines, were also relevant in vivo in the tumor tissue samples. For that, we used the gene expression, the transcriptome expression of the Cancer Genome Atlas for prostate adenocarcinoma, and we compared, we saw if those samples that have higher levels of methylation of non-coding 886 versus those that have lower levels of methylation of non-coding 886 were somehow associated to the expression of genes involved in cell proliferation. Since there is a proliferative gene signature that has clinical use with an approved clinical use in prostate cancer and is called Prolaris, we actually analyzed specifically the genes of this signature for its proven clinical value. So what you see is that when you analyze these patients, when you cluster the patients with high and low methylation of non-coding 886, based on the genes in the Prolaris signature, they group in the patient with a high methylation, meaning low expression of non-coding 886, have a higher expression of the proliferative signature compared to those who having a low expression or high methylation, a high expression of low methylation of the gene. With that analysis, we kind of validate that the proliferative genes are associated to non-coding 886 expression in the tissues of the patients. And during the course of our study, there were several papers coming out in different tissues, not in prostate cancer, but in other cancers, proposing mostly tumor suppressor gene roles of non-coding 886 as a whole molecule, but also oncogenic roles of this molecule, meaning that probably non-coding 886 effect in cancer depends strongly on the tissue type. So it's not a general tumor suppressor, it depends on the tissue type. So it's worth to further investigation, an investigation focused on the specific tissue type. So this is just a review of the literature in which cancers this Volt RNA has been associated to tumor suppressor or oncogenic function. So there's been, since the literature has been growing so much, there's many theories in also molecular pathways, different molecular pathways have been proposed for Volt RNA 2.1 in cancer tissue. Some of them associated to TGF-beta pathway, some of them associated to NF-kappa beta pathway, Dicer, JAK-STAT pathway, and a transcriptomic change that we still don't know the final effect. So it's an open field. So many groups are working in a specific hypothesis, but constantly people are discovering new pathways in which Volt RNA 2.1 is involved. So our next question was if Volt RNA 2.1, non-coordinate 86, could also be a microRNA precursor. So remember that initially it was classified a microRNA precursor, and potentially as any microRNA precursor, it could give rise to two microRNA complementary, the one derived from the 3' arm or the one derived from the 5' arm. So the one we have detected initially was the 3', yeah, this one, okay. So we made a little bit, what are associated with any? Yeah, that's exactly, thank you. Someone is asking if it's associated with our organoid, and that's, I hope I will answer your question, thank you. So here it is, the Volt RNA, canonical Volt RNA should be cut, clipped by Dicer, producing nice neat cuts, particularly at the 5' end of the microRNA. So if Volt RNA 2.1 produces a small RNAs similar to microRNA, we expect to see reads that nicely pile up in the region corresponding to the microRNA. If particularly it needs to be nicely piled in the 5' side. And that's what we see for the 3' UTR. Analyzing the small transcriptome data from many studies, including the TCGA, we found that there's a nice piling, therefore a nice processing, a specific processing of the Volt RNA in small RNA. And actually we see piling at the 3', but also at the 5' UTR. And we don't see many other piling. Sorry, this is not ours. We did that in prostate later on. So for the pattern of reads seen in the small RNA data, it seems that Volt RNA 2.1 produces two small specific RNAs of the size of microRNAs. This is another way to see. This is the Volt RNA. This is the 5' derived small RNA and the 3' derived small RNA. So this is what happened in the normal cell lines. Yes, you see nicely that. And then in the tumor cell lines, there was a phenomenon that I don't have time to discuss. We are investigating that now, but there is a change, a reduction in the expression of 3' and an increase in the expression of the 5'. We call that an arm switch preference processing that happens in the tumor. But I don't want to mess up this because it's already being dense. So I just want to tell you that for the pattern of expression of reads in the small RNA data in prostate cancer, analyzed in many data sets, including the TCGA, the evidence indicate that this RNA is processed specifically in small RNA. That means, well, maybe it's a microRNA precursor. Yet, as someone asked, this is also in evidence in other cell lines. Moving to the next one. As someone asked, there's microRNAs act as mRNA repressors by binding specifically to organoid proteins, particularly organoid 2. So if these small RNAs function as microRNA, they should be associated with organoid proteins. They should be associated to at least part of the microRNA pathway, the downstream part. So what we did was to reanalyze data from knockout of the microRNA processing. Yeah, we don't know if it's segregated in tumors. We know there's less amount in tumors. If it is for degradation or if it is for the processing, we don't know. I'm answering a chat question. Thank you for the question. Interesting one. We don't know. So what we did is we use that. We know that we see if this microRNA is processed by DROSHA, we should see that the knockout of DROSHA will reduce the amount of the microRNA. Again, if it's exported by exportin 5, the same thing. If it's actually processed by DICER, it should be an effect on the expression of the microRNA in the knockout. So if you look at that, and again, I cannot go into detail, but this is, you see, and when there is a knockout of DROSHA, you don't see a change, even though this is a change in the global amount of data. It's the amount of microRNA remains stable, and that is because it's increased relative to the total, because the other microRNAs are reduced. But then it's same thing with 5P, and with a microRNA that is not processed by DROSHA. Not all the microRNAs are processed by DROSHA, as I said before. Then when we look at the knockout of exportin 5 and DICER, it indicates that exportin 5 is not associated, but DICER knockout affects the expression of the microRNA. Sorry, I just need to be fast. And then about the association of the AGO protein, what we did was a reanalysis of the PARP clip experiments done in prostate cancer cell lines, when they see by the RNA-seq the association of small RNAs to Argonaute. And then what you see is that there's an association of microRNA 3P to the Argonaute fraction, but not much of this 5P. So that supports a microRNA action of the 3P, because it's associated, it's dependent on DICER, and it's also associated with Argonaute protein. These are controls that you can look at later. microRNA 3P is not lowly expressed, it's over the average expression of the microRNA, so its amount also suggests a function in the cell. So by analyzing many different data sets, we confirm that there's a downregulation of the expression of this small RNA 3P in many prostate data sets. When we looked at our small cohort of patients, we also see by qRT-PCR a decrease in the expression of the microRNA from the normal to the malignant tissue in matched samples. Same thing happening in the cell lines. There's a reduction of the expression of the microRNA assessed by qRT-PCR. I'm going fast. The cell lines, some of them have high, some of them have low methylation. There is also a correlation between the methylation and the DNA methylation of the gene and the production of the amount of 3P and the amount of the precursor. So what we think is that the methylation of the promoter affects the expression of the Volt RNA as a whole molecule, and that is also affecting the small RNA production. So the change in the amount of 3P is dependent on the change of the expression of the precursor molecule. Then there might be other processing events happening. Some of them may be accounting for this change in the arm switch expression that we are seeing in cancer, where 3p goes down in cancer, whereas 5p goes up in cancer. So one thing is the global amount of expression of these small RNAs. The other thing is the arm switch that we see also in cancer. Then we look in particular at the proliferative phenotype potentially caused by these small RNAs. Since we knew that the precursor affected the proliferation of the cells, we focus on that. Doing the transient transfection of the microRNAs, overexpressing the microRNAs, we analyzed firstly the gene expression changes, and we saw that the genes that are downregulated in the experiment have an important enrichment, a significant enrichment in the seed of Mir886 or non-coding, small non-coding 886-3p, meaning that this small molecule is reducing the amount of mRNAs bearing a complementary sequence of its seed in the 3' UTR, and that's also an evidence of a microRNA-like function for this small RNA. We confirmed these arrays in the U145 cell line, LNCAP, and PC3 for several genes. Some genes were during the course of this study identified as direct targets of the small RNA in other tissues, including pol-like kinase 1, TGF-beta 1, CCD6, CDT1. So among these genes that are repressed by the transfection of small non-coding 883P, there are at least 4 genes that have been validated as direct targets in other tissues, which indicate that these analyses were biologically relevant and again support a microRNA function for this microRNA. So when we look at the enrichment in biological functions of these genes downregulated by 883P, one of the pathways that comes up is apoptosis and cell cycle. And as I said before, we already knew that proliferation was affected by the precursor, and so it may be part of the reason is that the microRNA is working in this proliferative change. So we look at the proliferation of the transient transfection of the microRNA, and we saw that this overexpression causes a decrease in the proliferation in different cell lines. We did that again by complementary cell cycle analysis, and we confirmed the phenotype in different cell lines. I'm not going into detail, but the overexpression of 3p then reduces cell viability, and that could be due to alterations in the cell cycle and in the modulation of apoptosis. So we think with the microarray data and also the genes that we found associated to organelle and have this complementary seed of cis nucleotides, we filtered a list with these 3 criteria of potentially direct candidate target genes of small non-coding 883P. And we analyzed the expression again of this list of potential candidate direct targets in the Cancer Genome Atlas prostate cancer adenocarcinoma cohort. And what you see is when you look at those genes, they associate clearly with the clinical characteristics, meaning that the overexpression is this microRNA is a tumor suppressor, So if it's the clinical characteristics of the patient who have a decreased levels of the microRNA are more aggressive. See, you see here that the association. Then when you look at these, particularly these signatures in the survival of the patient, what we see is there is a change, a significant change in the survival of the patient, depending on the expression of the potentially direct candidate genes of 886.3p. Yeah. So if the expression of the genes is higher, then the survival is lower, and that's more important or more stronger for the cluster of 106 genes that is associated in here. So that means that the genes that are potentially regulated by 886.3p are relevant for prostate cancer. But so during the course of the study, in other cancers, there were many reports of small non-coding 886.3p functioning as a microRNA precursor. Here, this is the list of articles in different tissue types, proving or showing evidence in favor of a microRNA-like function of this small RNA. So our contribution is specifically in prostate cancer. Other studies have been done in others. So it means right now that our hypothesis is supported by the literature. What we think now is this precursor molecule, VoltRNA to 1, this VoltRNA or non-coding 86 is also a precursor of small RNAs that function as microRNAs. We don't use the microRNA denomination because we think it's more appropriate to say small non-coding RNA. And what happened, in our opinion, in the prostate cancer evolution is at the beginning you have this expression in the normal tissue of the precursor and the small RNA, and through the neoplastic transformation you end up in a reduced amount of both molecules. This when we experimentally increase the expression of both molecules, we see a change in cell viability, cell invasion, tumor growth in vivo, and alterations in cell cycle, as I showed. When we overexpress the small RNA alone, we also see the reduction in cell viability and increase in apoptosis and alteration of the cell cycle. We think that then in the normal tissue, there's an expression of this VoltRNA that turns to get silenced by DNA methylation in the neoplastic tissue, leading to a reduction in the expression of the transcript and then the reduction in the expression of the small RNAs too. And that means that if these are reduced, then the direct targets are be repressed and overexpressed, leading to an increase in cell proliferation. So that's our current model. We are working now in understanding this molecular mechanism that is causing this phenotype. We also are interested in seeing why there is also an arm switching, as I showed you before. MicroRNA 3p, the subject of our study, reduces the expression in a cancer. However, the complementary microRNA, the 5p here in red, increases its expression. Even though the precursor molecule reduces globally its expression, the arm switch is so intense that the 5p is increased. So that's also a very particular situation that is, in my opinion, is interesting to investigate in more detail. I said that the increase in the 5p. That's something we are following up. We have some preliminary result, but I don't have time to tell you what. I just wanted to let you know this finding because I want you to live today with an idea that RNAs are very, very complex, and they are evolving into many different functions, in also different roles with a tissue specificity that can be surprisingly variable. And for the end of the talk is just to invite you to read, if you fell in love with the Volt RNAs in this short talk, to take a look at our last publication. Is in the format of a preprint, which is currently submitted to publishing in a journal, but this is a pan-cancer chromatin analysis of the human Volt RNA genes that uncovers the association with cancer biology. In this study, we analyzed the DNA methylation of the promoters of the Volt RNAs, the 4 human Volt RNA, cancer-wide, in every single type of cancer. And for us, it's amazing that we found really very different clues of their action in cancer. Some of them, and most of them, support the available literature. There's no contradiction with the available literature, but since we know we don't know so much about these Volt RNAs, this analysis also throws a lot of new hypotheses about the role of the 4 Volt RNAs in different cancer types. So this is a field that needs a lot of study. There's many things that may be relevant for cancer biology, including in cancer immunology, which is a very nice and hot field right now in cancer. So I think Volt RNAs are gonna tell us many new stories in the next future. So hope if you are interested in the field, you can contact us or read our work, and also contact us for more details of our research. I think it's been already long enough. I apologize for the Spanglish and the Portuguese and these slides not in so many languages, but I hope you understand. But believe me, this is a complex subject, so many people can have ended confused. We were actually confused during the course of this research, and that was challenging, but also very exciting. And I think it's the same that is happening for other type of RNAs, including long non-codings, including tRNAs, including PIWI, etc. So we are just understanding the beginning of the multiple functions of the RNAs in the cells, RNAs and the byproducts. So thank you very much, and I don't know if we have time for any questions. Oh, sorry, I need to tell you that this is Rafael Fort. Sorry, Rafael is there. This is our group. Rafael and other collaborators, including Murillo, who participated in the first article. This is Rafael now. He defended the thesis. Now it's a joke. Last week, PhD thesis. So thank you very much. You are muted.
Muito obrigado, Mariana. That was a very nice talk. Acho que podemos falar português, né?
Sim.
Muito didática a sua apresentação, muito esclarecedora. Adorei a foto do Rafael, eu não imaginava que ele estava assim depois da defesa do doutorado. Isso é o que o PhD faz conosco, né? Muito bom. Nós temos algumas perguntas, acho que você já respondeu uma das perguntas ao vivo, né?
Sim.
Uma das perguntas foi aqui do próprio canal de pós-graduação. Acredito que seja o professor Michel, deve estar nos assistindo, aqui do Instituto de Biologia também. Ele perguntou se esse voo estava associado ao argonauta, e você mostrou para a gente que sim, né? E também fez uma outra pergunta aqui, se o braço 3P estava degradado em tumores, né? E acho que você quer responder, mas acho que você acabou falando já na sua apresentação, né? Talvez seja uma conjunção de duas coisas, né? A metilação mais o arm switch, né? Então, quer fazer algum comentário?
No, I think I tried to answer that at some point, that we don't know if it's a degradation of its processing, somehow processing with be degradation somehow, but it's not exactly the same, depending on what you mean. We don't know, and we are very interested in trying to find out. We have made some analysis, but we really don't know yet. We don't even know if microRNA 5P, this smaller 5P RNA, has any role in prostate cancer, even though there are several. There's a lot of consistency in the literature, because most of the reports about the role of this 8XE5P in other cancers is that they prove that it's an oncogenic small RNA. So, and that's exactly what our data suggests. Our data is only gene expression. We are just seeing the abundance of the microRNA. We don't know the effect. We haven't done any experiment about it. But it's interesting that the literature supports 100% oncogenic role of this 5P, whereas for the 3P, the literature in other solid tissues supports mostly, almost 100%, a tumor suppressor role. So we think is what we are going to find also. I like the second question that someone wrote. I don't know if the same person. What is the more determinant in cancer? Volt, RNA, the precursor molecule, or the microRNA derived? That's a very, very interesting question, and that's another future aim of our study, is to compare the action of both things, because so far we have looked at the expression of the precursor, which obviously produced the small RNA, and that's what happened in the tissue. The tissue two things are working together, right? And we dissect somehow the expression of the small RNA by transecting it individually, the small RNA, okay? But we still want to know exactly what the precursor does per se, right? Like blocking the microRNA. We haven't worked enough in that to tell you an answer, but that's something we would like to do. Also, but the other thing that I can tell you is that the expression of the precursor molecule is really higher than the expression of the small RNA, and that may tell, but I don't know, that the precursor has a function per se too, and also in line with the recent literature about the role of the Volt RNA 2.1. I didn't have time to talk about that because I think it was impossible for me to present all of this. But there's a recently open field on the regulation of the pattern recognition receptor, a bunch of RNA binding proteins that recognize double-stranded RNAs, highly structured RNAs that are involved in the native or the immune response. So, and we have also data from microarrays that I didn't have time to show here that confirm the same thing. So I believe that the RNA, the big one, the Volt RNA 2.1, has an important function in tumor immunity, and that's what we are trying to prove now. And I think the microRNA is likely doing something quite different, probably in working more specifically at pushing cell proliferation or some other cancer attributes. Hope I answered that.
Okay, that's an interesting topic.
An amazing question.
Yeah, we have another question here. Have you found evidences of intercellular communication using these RNAs?
Oh,
I love your question.
Yeah, that's something also that I couldn't show here, but we have also preliminary data about. So these small RNAs are secreted, and so we have done some reanalysis of exosome data in tissues, in different tissues, and exosome and vesicle associated particles released in the circulation. And yes, these molecules are in the extracellular compartment. We also have evidence that this Volt RNA, the overexpression of the Volt RNA that I showed you before, pushes a modulation of several genes involved in the exosomal pathway. So we are quite excited about that, and we think it's probably doing something, and also taking into account the modulation of the native immune system. We think, or we dream about, that's a lot of speculation, but we have a lot of evidence that can make the puzzle. Thinking that I think that this Volt RNA might be changing the secretome of the cell in part, sending immune-related signals to the environment, and also perhaps sending also small microRNAs derived from it in the shape of ribonuclear protein particles, or also exosomes, and somehow modulating the exosome pathway itself. Maybe.
Can you detect the precursor or the small RNA?
No, what is been seen so far is the small RNA. The problem is that I couldn't talk about that also, but sequencing of the middle-sized RNA is almost impossible, because that's why also these Volt RNAs are really still in the dark side of the genome, because they are not present in the typical transcriptome studies, because we study small RNAs by cutting RNAs of a certain size, usually no more than 35 nucleotides, and then we cut the transcriptome, the long RNAs, because of size or because of polyadenylation, depending on the strategy. But usually there's a size selection too. So the middle size actually RNAs are like a new field because of that reason, because there's not much knowledge about them, their expression. Also, because they are highly structured, they are very difficult to sequence by the typical RNA-seq approaches. So that's why they have remained in the darkness so far. And that's why we need to infer the expression from, in this case, methylation, or from the small RNAs produced or derived from them.
Okay. What about the copy number? What about the copy number? They are transcribed at low levels. The copy number of the transcripts.
The transcript copy number, I think I have it on a one.
They are highly transcribed.
They medium highly transcribed. It's only one locus. Locus is only one, and it's medium high transcription. Yeah, it's ubiquitous.
Okay, very nice, very interesting.
Well, so I'm glad they have done the right questions. I don't know if I thought I wasn't clear enough, and I apologize for that. But I think the questions came up quite nicely. So at least some people, found the most important question. I hope they understood and they enjoyed the talk.
Okay.
Thank you for the invitation.
I appreciate your taking your time to have a conversation with us. E acredito que já estamos passando bastante do tempo, né? Mariana, muito obrigado em nome da pós-graduação aqui do Instituto de Biologia. Eu agradeço mais uma vez a sua disponibilidade.
Obrigada.
Meu parabéns ao trabalho de vocês. E eu encerro, então, a nossa sessão. Obrigado a todos os espectadores pelas perguntas também. Um abraço a todos e até breve.
Bye, bye.